Hello everyone, I'm Tania Tamara Pedrozo and I work in the Instituto de Biotecnología de Misiones Dra. María de BRK, dependent of the Facultad de Ciencias Exactas, Químicas y Naturales of the Universidad Nacional de Misiones. I'm going to present my work titled Molecular and Phylogenetic Identification of two native biocontrol isolates from Misiones province, Clonostachys HEP30 and Paecilomyces HEP24. Okay. Chemical pesticides traditionally used for pest control may have negative consequences for the environment and the human population. So, as the alternative, the use of microorganisms or the metabolic products as biological control agents is a promising strategy. Among the microorganisms with biocontrol capacity, we find viruses, bacteria, and fungi. Particularly, the fungal mechanisms of invasion, persistence, and propagation characterize them as excellent biocontrol agents. In this case, the correct identification of biotechnological potential fungal species in a bioproduct is a prerequisite for any biocontrol strategy. However, the macro-micromorphological characterization present limitations, since the morphological characteristics are subject to variations in environmental conditions and at the time of the fungal life cycle. In this context, molecular identification allows overcoming these limitations. Therefore, it was proposed as objective of this work, molecularly and phylogenetically identify two fungal isolates with biocontrol capacity, Clonostachys HEP30 and Paecilomyces HEP24. So, first of all, both isolates were reactivated in petri dishes with potato destruct agars as culture medium, from where we got the mycelium to extract the DNA. Subsequently, the reason ITS1 5.8S and ITS2 was amplified and sequences. Consensus content sequences was constructed using the GNAU's PRIME program, and the analysis of identity and similarity were made with two databases. One primary database of the NCBI using the nucleotic blast tool, and with a secondary database of fungal barcoding, which is specific for fungal organisms with a pyruvate alignment tool. So, to generate a greater robustness to the local alignment of the isolate sequences, a phylogenetic analysis was performed using the MEGA program by three methods: neighbor zoning, maximum likelihood, and maximum parsimony. As the result of this analysis, we got that in the analysis of similarity with both databases, the isolate Clonostachys HEP30 presented high percentages of identity, up to 98% with Clonostachys Pityroides species. With the same analysis with both databases, the isolate Paecilomyces HEP24 also presented high percentages of identity, up to 98% with Paecilomyces variety species, as we can see here. Secondly, in the phylogenetic analysis with the three methods, allowed the separation of the species Clonostachys and Paecilomyces genera into different monophyletic clades with high bootstrap values up to superior to 90%. As we can see here, we have a extract of the neighbor zoning method for better visualization. In this way, the Clonostachys HEP30 isolate clustered within the monophyletic clade of the Clonostachys Pityroides species, and the Paecilomyces HEP24 isolate was clustered within the monophyletic clade of Paecilomyces variety species. Then we can conclude that the amplification of the ITS1, 5.8S, ITS2 region, the molecular characterization, the construction of phylogenetic trees, and the conformation of monophyletic groups allow the molecular identification of the Clonostachys HEP30 biocontrol isolate as belonging to the Clonostachys Pityroides species, and the Paecilomyces HEP24 biocontrol isolate as belonging to the Paecilomyces variety species. Okay, thank you so much for your attention.